My name is Celine Berthier, and I am Associate Research Scientist in the Michigan Kidney Translational Medicine Center at the University of Michigan. In this short recording, I will present a new kidney disease model that I have developed with my colleagues Akihiro Minakawa and Jennifer Harder. Chronic kidney disease is a worldwide problem, but factors that contribute to development and progression of disease are inadequately understood. It is thought that kidney hypoxia contributes to the development and progression of kidney disease, but this is challenging to investigate as hypoxia is a normal physiologic state for part of the kidney. Hypoxia can occur under various conditions and is characterized by a decrease of tissue cell oxygenation, to which cells quickly respond by modifying processes involved in transcriptional and protein production and post-translational modifications. This often leads to tissue injury, which can be irreversible. The hypoxia inducible factor, or HIF, is a key player in this process, and specifically the HIF-1 alpha member. As described in a recent review from Kolsch, Dell, Rad, and Hall, kidney organoids are three-dimensional in vitro kidney culture models that can be generated from human stem cells. Kidney organoids demonstrate typical kidney tissue architecture and contain multiple kidney cell types. Kidney organoids have been shown to be useful for mechanistic studies, drug screening, and disease modeling. The aim of this study was to develop an in vitro hypoxic kidney organoid model and to determine if it recapitulates the hypoxia features observed in kidney disease, including the associated molecular mechanisms. I hope to convince you that the integration of complementary state-of-the-art technologies, computational biology tools, and functional studies is critical for studying human diseases. We generated kidney organoids from human pluripotent stem cells and cultured them under hypoxic conditions for 24 hours. Our goal was then to explore hypoxia-induced alterations in cell type-specific gene expression. I'm not going to describe the details here, but I'll be available for questions. The first step was to verify that the model developed work by measuring markers of hypoxia at both transcriptional and protein expression levels. Under hypoxic conditions, kidney organoids showed nuclear accumulation of HIF-1 alpha detected by immunofluorescence and increased expression of known HIF-1 target genes as measured by real-time PCR and ELISA. We also performed a functional study called the metabolic flux analysis using 13C-glucose, which confirmed that hypoxic organoids preferentially used glycolysis over oxidative phosphorylation. These findings demonstrate that our hypoxic kidney organoid model recapitulates gene expression and cell function expected in hypoxic kidney tissue. To better understand the development and progression of kidney diseases, it is critical to evaluate how each specific kidney cell type reacts to injury. To pursue this goal, we analyzed both bulk, mRNA, and single-cell transcriptomic data from hypoxic kidney organoids. Single-cell data were integrated by fast integration using a reciprocal PCA using Surrey version 4 with a minimum of 500 genes and a maximum of 5,000 genes expressed per cell, and less than 50% of mitochondrial reads per cell. 10 cell type clusters were identified and analyzed using the cell by gene tool. Differential expression analysis comparing hypoxic to normoxic organoids revealed increased expression of HIF-1 alpha and HIF-1 alpha targets in podocytes, stromal, proximal tubular, and distal tubular cells. Expression of genes involved in key metabolic pathways, including glycolysis and mitochondrial functions, was affected, most notably in podocytes. Causal network inference analysis confirmed HIF-1 alpha as a top upstream regulator of the observed bulk and single-cell transcription responses. Proteins corresponding to the decreased mitochondrial transcript seen under hypoxia conditions in kidney-specific cell types were also affected, as detected by immunofluorescence staining of the organoids. In summary, our hypoxic kidney organoid model recapitulates key events in hypoxic kidney tissue. Hypoxia resulted in nuclear accumulation of HIF-1 alpha, increased transcription and protein levels of known HIF-1 targets, and decreased expression of key mitochondrial genes necessary for efficient ATP generation. And indeed, a functional analysis of glucose metabolism demonstrated that glycolysis increased and oxidative phosphorylation decreased in organoids exposed to a hypoxic environment. Further, single-cell RNA sequencing analysis showed that the hypoxia-induced transcriptional alterations were occurring in kidney cell types. Interestingly, alterations in mitochondrial gene expressions were the most profound in podocytes, a key cell type involved in kidney filter function, suggesting hypoxia could contribute to cell type-specific injury. In conclusion, Computational analysis integrating multiple omics datasets demonstrate that hypoxic kidney organoids capture key metabolic pathway perturbations seen in kidney disease. In combination with protein expression and functional studies, these data demonstrate the relevance of the hypoxic kidney organoid model to study pathomechanisms in kidney disease. Thank you.